Okay, good morning, students.
Good morning, ma'am.
Good morning. So, I'm going to start the class today. I'm going to take on chromatography. So, kindly answer back, okay, if I ask any questions. So, chromatography, from the word itself, chromatography, it means writing in color. Okay, the word chroma means color and graphy means to write. So, this chromatography, it was invented by this man known as Michael Sweat. He was born in Italy, but he is a Russian botanist. So he was the one who invented this chromatography while doing experiments on plants to separate chlorophylls and carotenoids. So he developed this technique. Okay. So chromatography, it is a technique that is used in the lab in which we separate closely related compounds from a mixture. It is a physical method of separation. Okay, and the principle is based on two phases. One is the stationary phase and the mobile phase. Okay, so in chromatography, the principle is based on these two phases, stationary phase and the mobile phase. What happens is the stationary phase will be there, and in the mobile phase, this the solute or the analyte that we want to separate, what it will do is it will be distributed between this stationary phase and this mobile phase. So this chromatography, it is based on the physical method of separation where the components of this sample mixture that contains a sample that we want to analyze, this will be separated as a result of their differential distribution between the stationary and the mobile phase. It will be more clear in the coming slides, okay? So before going to in detail about chromatography, let us learn about some chromatography terms. Number one is a chromatograph. What is a chromatograph? This is an equipment that enables a sophisticated separation. Then what is eluent? Eluent is the fluid that will enter the column, or it is the solvent that will carry the analyte. Then what is eluate? Eluate will be the mobile phase that will be leaving the column. And stationary phase. Stationary, from the name itself, it is an immobilized phase. So this stationary phase will be immobilized on the support particles or on the inner wall of the column tubing. We have silica layer that we use in thin layer chromatography, and we have methyl silicone polymers that we use in gas or liquid chromatography. Then what is mobile phase? So mobile phase is the gas or liquid. It can be either gas or liquid which will percolate or which will enter through or along the stationary bed in a definite direction. Then what is the retention time? Retention time is the time taken for a particular analyte to pass through the system. So you'll come across these terms again and again. Now, what is the purpose of chromatography? Why do we need this technique? It is mainly for two purposes. One is analytical and another one is preparative. Analytical will help us in determining the chemical composition of a sample. Okay, whereas preparative means we require those substances to purify and collect one or more components of the sample. Okay, so there are two purposes of chromatography. One is analytical and another is preparative. Now, what are the various uses of chromatography? So some of the uses of chromatography are in pharmaceutical company to determine the amount of each chemical that is found in the new product. Then in hospitals, to detect blood or alcohol levels in the patient's blood, okay, to detect the various analytes that you want to know. Then in forensic or law enforcement, to compare a sample found at the crime scene to samples from the suspects. Then in environmental agency to determine the level of pollutants in the water supply, and also in manufacturing plant to purify a chemical needed to make a product. So what are the various forms of chromatography? We have planar chromatography and column chromatography. So planar chromatography is of two types, paper chromatography and thin layer chromatography. So what is paper chromatography? This can come in your exam, paper chromatography. This is one of the most common types of chromatography. Okay, here we use a strip of paper that is made of cellulose, and by capillary action it is used to pull the solvents up through the paper to separate the solutes. Whereas in thin layer chromatography, we use another absorbent material on a flat glass or a plastic plate. So this paper chromatography, as I told you, the paper is made of cellulose, okay? And we have a solvent system that is made of butanol, acetic acid and water in the ratio of 4:1:1. Now you have to remember, here in paper chromatography, we are using a paper made of cellulose. This is a supporting medium. It is not the stationary phase. Okay? Here the stationary phase will be the water molecule that will be held between the cellulose fibers. Remember, the stationary phase is not the paper, but the paper is just a supporting medium, whereas stationary phase is the water molecule that will be held in between the cellulose fibers of the paper. Then mobile phase is less polar. It will be acetic acid and the butanol. Now paper chromatography is again of two types, ascending and descending. So in ascending chromatography, from the name itself, okay, you can understand. So it is ascending. Okay, if you see this tank here, so we have this tank here, okay, then this is the paper, this here is the solvent. So before we put the paper inside this tank, what we do is we have to mark the paper, okay, about 2 centimeters from the edges, okay. We have to mark a line here with a pencil. We have to be careful while doing this experiment and not to touch with our hands in other places of the paper, okay, because there will be some some pigments or some other materials in our finger, and that will leave a mark when you stain it later. So what you do is you draw a line with a pencil, and then you mark the points where you want to put the samples. Okay, for instance, here we have 3 points here where you put the sample. So just a small drop, not too much, just a small drop of the sample you apply on those points. Then after that, we put this paper in this tank which contains the solvent, okay, and then we cover the tank and we leave it for about 14 to 18 hours. So what will happen is, in that time, this solvent, okay, it contains the mobile phase, and the stationary phase is the water molecule. So this will be carried up like this, okay, and then we should make sure that this solvent should not go beyond the edges of this paper. Just about 2 centimeters or 1 centimeters before it touches this edge, we should take it out. Usually it will take about 14 to 18 hours for this procedure to be complete, okay. Then after that, we take this out, we take the paper out, okay, and then we dry it. After drying, then we will stain it, okay. After drying, then we stain it. When we stain it, we will get the different colors accordingly, where that particle, that analyte which we want to check, that will be carried by the solvent, okay. So we will get different colors where that analyte is carried away. Okay, so that was for ascending chromatography. Then in case of descending chromatography, what we do is the paper, we have to apply it's the same thing. When we draw the line, then we apply the sample, but here it is in descending order. Okay, so here we have a paper support system, and we have a trough, and in that trough we will fill it with the solvent system. Okay, then we will leave it for 14 to 18 hours, and that solvent system will flow down in descending order. Okay, and in the same way we will take this out, dry it and stain it. So coming to another term known as partition coefficient. What is partition coefficient? It is nothing but distribution of the solute in stationary phase by distribution of the solute in the mobile phase. And another one is distribution coefficient. This is concentration of component in stationary phase and concentration of component in mobile phase. The only difference between these two is that in case of the partition coefficient and distribution coefficient, the difference is just that in distribution coefficient it is mainly for the ionized molecules, whereas in partition coefficient it can be of any, okay, ionized and unionized also. So for separation of amino acids, if we do it by paper chromatography, we will require the cellulose fibers of this sheet of chromatographic paper. As I told you, this is the supporting media. Then we will require the solvent, okay, then cellulose, the beakers, and the stain that we use is ninhydrin in 1% butanol. Now, what are the various factors that will affect this migration? This depends on the solubility of the solute in solvent, the intermolecular forces, also the pore sizes of the filter paper, and the size of the solute. Now again we'll come across two important terms. One is the solute front and one is the solvent front. So what is this solute front and what is the solvent front? So suppose this is your point of application, okay, of the samples. Then after you take it out, this paper will still be wet, okay. So this paper will still be wet. Then you mark that end where the paper is wet, okay. You have to draw a line with a pencil. So from the point, from this edge till the end where the solvent has passed, that is the solvent front. And from the point of application of the sample till wherever the sample is stained is located, that is the solute front. So another important term is the retention factor. So what is this retention factor? It is just distance traveled by the component. See here in this picture, here is the sample spot where we applied it. And at the end of the experiment, we got this sample, it has traveled till here. We got a stain here. So this is the solute front. And this is the end where the solvent passes. So this is the solvent front. So the retention factor is just the distance traveled by the component divided by the distance traveled by the solvent. That is A divided by B. Okay. So knowing the retention factor of this particular sample, we can then identify this sample. This is just the same, okay. So this is, suppose here is the origin of the samples, and at the end of the experiment, when we stain it with 1% ninhydrin, we get these different spots where the sample has been carried away. So the retention factor will be the distance traveled by this component. For this particular sample, it will be the distance traveled by this component divided by the solvent front. It will be different for these different samples. So by knowing that retention factor, we will be able to identify the amino acids. Now, if the retention factor is 0, what does that imply? It means that the solute remains in the stationary phase. It is immobile. If it is 0, that means this solute, it would be here only in the origin. Okay, so that means it is immobile. And if it is 1, that means the solute has no affinity for the stationary phase, and it also travels with the solvent front. If it is here, then it will be A divided by B, which is 1. So that means it has traveled the same distance as traveled by the solvent front. So that is the importance of retention factor, which is also known as relative front. So what are the factors that will affect this retention factor? It will be the solvent system and its composition, the quality of the solvent system, quality of the paper, direction of fibers in the paper, and temperature. So all these factors will affect this RF value. That is why when we perform this experiment, we perform it in a closed system. Okay, this tank should be closed after we have kept this paper inside the tank. So these are some of the values of the retention factor values of some amino acids. So when we perform the test, when we calculate the retention factor value, when we get whatever values we get, we can compare it with this, and then we will be able to identify the respective amino acids. Most of the amino acids, after staining, it will be purple in color, whereas for proline and hydroxyproline, it will be yellow in color. So that was for paper chromatography. Then we have something known as thin layer chromatography. This is also quite similar, but in this case we use a very thin uniform layer of silica gel or alumina, and this we coat it on a piece of glass, metal, or a rigid plastic. So this silica gel or alumina, we made a slurry of it and we coat it into this glass, metal, or rigid plastic. And here the stationary phase is the silica gel or the alumina, and the mobile phase will be a liquid solvent or a mixture of solvents. How will the compound move? They will move by capillary action. So this is an example of thin layer chromatography. It is very small, okay, this thin layer chromatography glass plate, this is coated with the silica gel. It is very similar to this paper chromatography, but there are various advantages of this thin layer chromatography. Number one is it takes lesser duration to complete than paper chromatography. So it is the same way only. You draw a line where you will apply the sample, okay, and then you put it inside a beaker. You put this paper or the rigid plastic that is coated with the silica gel or alumina gel, and you put it inside this beaker, okay. And in the same principle that occurs in paper chromatography, this also, the solvent will be kept in the beaker and the solvent will carry this mixture, okay, and before it passes through this, okay, about 1 to 2 centimeters, you should stop and take it out, okay. Now most of the time if you use organic compounds, they are colorless. So what they do is, after they take this out, they will observe the fluorescence under a UV light, okay. So the advantages of thin layer chromatography are greater sharpness of separation, then short development time, small volumes of sample are required, it is also easy to use, and we can do multiple analysis. However, there are also several disadvantages. Number one is it is not very quantitative. Then the plate length is limited. It is very small compared to paper chromatography. And separation takes place up to a certain limit. And this also occurs in an open system. So humidity and temperature may affect the mobile phase. So how do we prepare this thin layer chromatography plate? The silica gel or aluminium oxide, about 2 grams per 20 ml of the solvent, we have to make them into a slurry and then pour them on the glass plate. So we can add ethanol or methanol with the silica gel, and then we pour them on the glass plate and spread it evenly. And then we dry the plate at 100 degrees Celsius to remove the water molecules. The solvent system can be different, okay? It is not necessary that it has to be only the butanol, acetic acid and water. So in case of thin layer chromatography, if you want to separate amino acids, the stationary phase we use is silica gel, and the solvent we can use is this BAW, butanol, acetic acid and water. If we want to separate triglycerides, we use petroleum ether, diethyl ether and acetic acid. If we want to separate phospholipids, we use chloroform, methanol and water. So different solvent system for different compounds that we want to separate. This is just another picture of a thin layer chromatography. Okay. So here is the thin layer plate with silica gel surface. Here we have load the sample. Okay. This is a beaker and we put this solvent in the beaker. Then we take this thin layer plate and we put it inside. So we allow it to develop, and at the end we will stain it and we will be able to observe these different samples that were present in the mixture. So what are the various stains that we use? In case of amino acids and proteins, we use ninhydrin in alcohol. Then in case of lipids, we use rhodamin. In case of phospholipids, we use sulfuric acid. In case of steroids and terpenoids, we use antimony trichloride. Then ethidium bromide we use with DNA samples. And for sugars and carbohydrates, we use diphenylamine. So that is for the planar chromatography, the paper chromatography and thin layer chromatography. Now there is another important one, column chromatography. This is another form of chromatography. In column chromatography, we use this column that can be made of stainless steel or this plastics. Okay, so column chromatography, the principle will be same. We will have the stationary phase and the mobile phase. Now you can see here in this diagram, this is the stationary phase and here this is the loaded sample. So what is important is that the sample to be separated must be kept at the highest point of the column. Okay, and then that sample will go through the column at various rates, and that substance will be absorbed by the stationary phase, and depending on their differential distribution and on the forces that occurs between the mobile phase and the stationary phase, the samples will flow down at different speeds, and then we can collect the different sample. So as you can see here, this column, we take it, okay, and then we put a stationary phase. Stationary phase, let's say we put here silica gel or alumina gel, okay. Then after that, we load the sample. We load the sample, then we pour the mobile phase. When we pour the mobile phase, if you can see here, the sample has separated here, green and yellow. Green and yellow here. So this green color sample, it has weaker interaction with the stationary phase. So it will flow faster. Whereas this yellow color sample, it has stronger interaction with the stationary phase. So it will flow at a lower rate. Okay. Then as this green color will come down, then we can collect it, and then separately this green color and then this yellow color. So the stationary phase in column chromatography can be made of pure silica, cellulose or dextran. And this can be packed into the tube, or it can be coated on the inner surface of the tube. Okay. So what are the various types of column chromatography? We have adsorption chromatography, partition chromatography, gel column chromatography, ion exchange column chromatography, gas chromatography, and high performance liquid chromatography. So the name, it can be liquid chromatography or gas chromatography. Now, why do we say it as liquid chromatography or gas chromatography? Depending on the mobile phase. If the mobile phase is in liquid form, it is known as liquid chromatography. And if the mobile phase is in gaseous form, it is known as gas chromatography. These names are derived based on their mechanism of separation: partition, adsorption, size exclusion, ion exchange, and affinity chromatography. So, partition chromatography. This is used in both gas chromatography and liquid chromatography. We have discussed about paper chromatography. That is one of the example of partition chromatography. Here, the differential distribution of solutes between the two liquids, that is the mobile phase and the stationary phase. That is the basis for the separation. So if you can see in this diagram, this black dots, these are the solute that are dissolved in the liquid phase, coated on the surface of the solid support. So in case of paper chromatography, the solid support is the paper made of cellulose, but the mobile phase is the solvent, the butanol and acetic acid, whereas the stationary phase is the water. So based on the differential distribution of the solute between the water and the solvent, that is the basis of separation. So the molecules will partition into the stationary phase based upon the affinity for stationary phase, and eventually it will partition into the mobile phase again. So the ones that will have a stronger bond with the mobile phase, which is polar, that will flow slower. Okay, it will be retained in the paper earlier. Then in case of thin layer chromatography, a thin layer is coated onto inside of thin layer is coated onto inside of gas chromatography column or on small particles on liquid chromatography column. So partition chromatography can be used in both gas chromatography or in liquid chromatography. And as I told you, the name gas chromatography or liquid chromatography, it is based on the mobile phase and not on the stationary phase. Then another is adsorption chromatography. From the name itself, you can understand it is based on the principle of adsorption. This is very similar to partition chromatography. So the solutes will be adsorbed on the solid particle, and how it will be removed or desorption, removal of the molecules, is known as desorption. So this adsorption will be based on various forces such as electrostatic force, hydrogen bonding, and van der Waals forces. These forces will control the adsorption. So the stationary phase can be silica, alumina, or powdered charcoal. Okay, and then the mobile phase will be liquid, and the sample mixture you have poured, so some of the solute will be adsorbed, and those that will be desorbed will flow faster. So you can see here in this column chromatography, so this is the stationary phase. So we have applied the mixture containing X and Y components. Then we have applied the mobile phase. So now this mobile phase will flow down and it will carry this mixture through this stationary phase. And while this mixture is passing down through this stationary phase, the molecules will be separated. And for example, here you can see these two colors, pink and green. So the substance, suppose this green is the Y, it is not adsorbed, whereas this X is adsorbed in the stationary phase. So it will be coming down much more slower than this Y, which is not adsorbed to the stationary phase. Then we will collect them accordingly. So that is adsorption chromatography, based on the principle of adsorption of the solute with the stationary phase. Then another is ion exchange chromatography. Ion exchange chromatography. This is the separation of ions, either cations or anions. And the principle is based on the relative strength of the ionic bond. So you see here in this column chromatography, here in the stationary phase, we have positively charged gel beads of charged diethyl aminoethyl cellulose. Okay, so this is the stationary phase. It is composed of this positively charged gel beads. Then we apply the sample mixture from the top. Now what happens is, in the sample mixture, those negative molecules will bind with the positively charged beads in the stationary phase, whereas the positive molecules, they will repel, they will not bind, so they will elute out first. Okay, and the negatively charged they bind to this positively charged beads. Now in order to remove them, what we do is we can pour a buffer solution or a solution containing a high salt concentration, and that will elute the negatively charged molecules and we can collect them. So this ion exchange chromatography, the principle is based on the relative strength of ionic bond, and so we can separate the cations or anions accordingly by using stationary phase, either made of positively charged beads or of negatively charged beads. Students, are you still there?
Yes, ma'am.
Okay, so another one is size exclusion chromatography. Okay, size exclusion chromatography. From the name itself, you can understand that the separation is based on the size. So here what we do is, this stationary phase, we use these beads, agarose beads. These beads, they contain pores. Okay, you can see here, these beads, they contain pores. Okay, so what happens is, when you put the sample mixture, the small molecules, they will enter through these pores. They will get trapped in these pores and they will take a longer time to get out. Whereas the larger molecules, they will easily come out fast. Okay, so you can see here in this picture here. So these are the resins or agarose beads, they contain pores. And when we apply the sample mixture, the red color here, they will enter through the pores inside the agarose beads. So they will take a longer time to flow down this stationary phase, whereas the larger molecules will come down fast. That is one. But another reason why this larger molecules, they come down fast also, it is due to the force of gravity. For example, if you stand on top of a building, you throw a stone, it is heavier, so it will fall down faster, whereas if you throw a feather, okay, that will take time to reach the ground, right? So that is based on the gravity. Okay, so we finished partition chromatography, then adsorption chromatography, ion exchange chromatography, and size exclusion chromatography. Now coming to affinity chromatography. Affinity chromatography is one of the techniques that is very selective, and the principle is based on specific molecular interactions. The various specific interactions are interaction of antibody with antigen, of enzyme with substrate, of a receptor with a ligand, and of lectins with sugar. This affinity chromatography, it is used to purify antibodies or other proteins that are used in research, and this is used a lot in biological applications. So the principle is based on the specific interactions between the antibody and antigen, or enzyme and substrate, receptor with ligand, or lectin with sugar. So now here, if you can see in this diagram, so the stationary phase here, it is composed of these agarose bead, and these agarose bead, they have a ligand. Okay. Now what we want to separate is some target protein. Okay. This protein that will bind with this ligand. So in the stationary phase, we have this agarose bead with this ligand. Okay. So this sample mixture, it contains the target protein and some other components. So when we add this sample mixture, this target protein, it will bind with the ligand. It will interact and bind with this ligand, whereas the other components will be eluted out. Okay. Then now, because what we want is this target protein. So this is bound to this ligand, but we want to this only. So what you do is, we have to apply an elution buffer. Okay. And this will then remove this target protein from this ligand, and now we can collect this target protein. So this is affinity chromatography. Whereas if you want to separate some enzymes, then this agarose bead will be attached to a specific substrate or to a specific enzyme that will bind with a specific substrate, or vice versa. So another important type of chromatography is the high performance liquid chromatography, or high pressure liquid chromatography. This is a type of liquid chromatography, but the principle can be different based on the mechanism of separation. It can be adsorption, ion exchange. Okay. Here in column chromatography, in the others, so you just let the mobile phase, and the sample mixture pass through the column slowly. Whereas in this high performance liquid chromatography, we use a very high pressure. Okay, and this will help us to perform this technique very fast. So before going further, let me just talk about chromatogram. What is this chromatogram? It is just a signal, a visual output. Okay. So like, ECG. So you put some leads on your chest, and then the output you get is a graph. Okay. The ECG. So it is very similar to that. It is just a visual output, and the x-axis here will be the retention time, whereas the y-axis will be the signal or the concentration, and these are the chromatographic peaks. Okay, so it is described in terms of the width, height, or the area. So this chromatographic peaks will be for different sample components. One will be for one sample component, two will be for another sample component. Okay. So chromatogram is nothing but an electronic signal that is plotted as a function of time, distance, or volume. So here in high performance liquid chromatography, in our lab we have one HPLC which we use to estimate this HbA1c. So if you want, you can come and check in the lab. So this is just a schematic diagram of HPLC. So here is the mobile phase in the reservoir. Then we have a pump. Then this we have this HPLC column. These are very small compared to the others. Okay, they are very small. And we have a detector. And this is the computer data section from where we can obtain the output or the results or the chromatogram. Okay. So the mechanism of HPLC is smaller particle size for the column packing material is used. So that will give us a much greater surface area. So the interactions between the stationary phase and the molecules will lead to a much better separation of the components. So this is the mechanism for HPLC. It can come as short notes, HPLC. Okay. Now what are the various applications of HPLC? in terms of our hospital application. Okay, one is investigation of congenital metabolic disorders such as maple syrup urine disease and phenylketonuria. So this ion exchange HPLC, it is more helpful in the separation and quantification of the different amino acids than paper or the thin layer chromatography. So this is much more important. As I told you, HPLC is high performance or high pressure liquid chromatography, but the principle can be different based on the different mechanisms of separation that we use. Okay, the column can be kept with the different cation or anion resins. Okay, so that will lead to will give us a principle of ion exchange HPLC. Okay, then another one is measurement of glycated hemoglobin. This is the one that we have in our lab. Okay, then analysis of biogenic amines. This is important in case of diagnosis of carcinoid syndrome or pheochromocytoma. Then in antenatal screening, then in case of porphyrias, then to study the levels of different vitamins and their metabolites like vitamin D. Okay. Now what is the advantages of HPLC? Number one, the speed, then its efficiency, accuracy. It is versatile and it is extremely precise also when it comes to identifying or quantifying the chemical components. However, this HPLC also has some drawbacks. It is very complex, and the most important drawback is of the cost. It is very costly also compared to the other forms of chromatography. Okay, so with this I have come to the end of this class. Can I ask some questions, students? So just to recapitulate, chromatography was invented by the Italian-Russian botanist Michael T. Sweat in the year 1900. Chromatography is a laboratory technique that deals with the separation of closely related compounds from a mixture. The principle is based on the differential distribution of the sample between the mobile phase and the stationary phase. The solutes or the sample with higher affinity for the stationary phase will stay longer than those with less affinity. Retention time, it is the time taken for a particular analyte. Analyte here is the sample or that we want to separate or we want to measure. So the time taken for that particular analyte to pass through the system is the retention time. And we have to choose the mobile and the stationary phase in such a way so that that particular analyte that we want to purify or we want to analyze will distribute itself between the two phases. So the main purpose of chromatography is to determine the chemical composition of the sample and to purify and collect one or more components of a sample for various other experiments. There are two important forms of chromatography, planar and column chromatography. Planar, we can divide it into paper and thin layer chromatography, whereas column chromatography can be liquid or gas chromatography. Based on the different mechanism of separation, column chromatography can be divided into partition chromatography, adsorption chromatography, ion exchange chromatography, size exclusion chromatography, and affinity chromatography. Whereas planar chromatography, in case of paper chromatography, we use a paper made of cellulose. But what you have to remember, which is very important, is that the paper is the supporting medium and water is the stationary phase, and the solvent made of butanol and acetic acid will be the mobile phase. Paper chromatography again can be divided into ascending and descending chromatography, paper chromatography, and you have to keep in mind that this should take place in a closed system. Okay, so what you have to remember is paper chromatography should be conducted in a closed system because temperature, humidity, all these and other dirt materials can affect the retention factor. What is retention factor? It is the distance traveled by the component divided by the distance traveled by the solvent. So when you know the retention factor of that sample, then you will be able to identify that sample. In case of amino acids, after you stain them in paper chromatography with 1% NEIGRIMS solution, most of the amino acids will be stained purple in color, like this, except for proline and hydroxyproline that will be stained yellow in color. And in thin layer chromatography, we use either a piece of glass, metal or a rigid plastic as the supporting media, but the stationary phase can be silica gel or alumina. The main advantage of thin layer chromatography is the short development time. This is the main advantage of thin layer chromatography over paper chromatography. Okay, so you please read up this one. So in your next practical, you will be asked questions regarding this topic. Okay, so the class is over for now.